So today we will be discussing about proteins. So what is actually protein? Protein is polymer of amino acids. Basically, these proteins are the long chains of amino. These are the long chains of amino acids basically, and these proteins are known as the building block. These are the building block of our body because they are responsible for the growth. They are responsible for the growth and the repair of our body. Repair of our body. These are responsible for the repair and the growth of our body, right? So these are the actually polymer of amino acids, right? Okay, now what are amino acids now? What are amino acids? Basically, amino acids are actually the monomers of the protein. Okay, actually basically our body is made up of, like, there are 170 amino acids which are found, 170 amino acids which are found, but our cells and our tissues but our cells and tissues are made up of 20 amino acids, are made up of 20 amino acids, right? Like in the whole, like, there are 170 amino acids which have been found, but our body is made up of only 20 amino acids, right? So now let's look at the basic structure of the amino acid, right? Okay, consider, like, this is the carbon, right? This is a group attached known as COOH, right, which is known as carboxylic group. Over here it is H. Over here we are having ammonium, and this is R. Okay, now this is known as carboxylic group, COOH, carboxylic group, right? This is hydrogen, this is amine group, right? This is known as amine group, and this is alkyl, right? This is alkyl. Anything can come in R, any kind of carbon chain, whatever do you want, you can adjust over here in R. And this is obviously the hydrogen. Okay, this carbon is known as alpha carbon, or it is known as chiral carbon. It is, if you're a student of chemistry, you must be known about the chiral carbon. This is chiral carbon. Okay, what does that mean? What is the meaning of chiral carbon? Okay, chiral carbon means a carbon in which every side has different group, like it is attached with different group. It is attached with different group, as you can see over here, right? Okay, this is another group, this is another group, this is another group, and this is another group, right? These four are having different groups. The carbon, the mid one carbon, the alpha carbon is attached with different groups. Here we are having COOH, here we are having H, here we are having NH2, and here it is R. So it means it is, the carbon is known as chiral carbon because it is attached with different kind of groups. Okay, all the amino acids are having chiral carbon except one amino acid, which is known as glycine. Okay, let's talk about the structure of glycine. It's quite easier. Okay, so this is a carbon. Okay, let me erase it. Okay, this is the carbon. This is like the chiral carbon or alpha carbon. Here it is COOH. Here we are having amine group, right? Here we are having H. Okay, in the position R, if I put H again, and this is known as glycine. It is known as glycine basically. So here this carbon is not, this is not chiral carbon. Why this is not chiral carbon? Why? Because you can see over here, this is H and H. What was the condition for chiral carbon? That the all four groups should be different, right? But over here, this is H and this is again H. So they are not different, like two are same and two are different. So it is not chiral carbon, neither it is alpha carbon. So all the 20 amino acids which are present in a body, from which our tissues and cells are having chiral carbon, except one, which is glycine, right? Okay, whosoever amino acid is known as, like, it has chiral carbon, it is optically active. That carbon is optically active. Now what does that mean, that they are optically active? That whenever you will pass light from them, the polarized light, right? If you have seen my videos of dextrose sugar, dextrorotatory, levorotatory, racemic sugar, you will quite understand this topic more clearly. If you haven't watched the video, go back and watch that video first. Okay, what does that mean, optically active? Actually, optically active means whenever you pass the polarized light from them, the light will be bended either in the right direction or in the left direction. If it bends to the right direction basically, so that's will be known as dextrose, right? And if it bends to left direction, it will be known as levorotatory, levotrose, right? So this carbon, whosoever compound is having this chiral carbon, they are optically active, right? But the compounds which is not having this chiral carbon, are not optically active. So it means all the amino acids from which our cells and tissues are made are optically active, except one, which is glycine, right? I hope it is clear to you. Okay, now actually these amino acids act as base and acid, right? Okay, consider like this is a jar, right? And it is having the neutral medium, and we have placed a amino acid over here, which is COOH, H, NH2 and R, right? So this is a neutral medium over here. What will happen over here, that H from this acid will move to NH2, and something, this structure will be formed, CH, COO minus, NH3 and R. This structure will be formed, and this is known as, like, this is acid. Now this is acid, and this NH3 is basically base. Like acid and base both are present. So now this amino acid is known as neutral amino acid. This is known as neutral amino acid, or you can say that it will behave as bipolar. It will behave as bipolar, right? Or you can say it is Zwitter ion. It is Zwitter ion. The amino acid which behaves as a neutral amino acid is known as Zwitter ion, right? Now next we are having, like, consider this is another container. There is another container, right? And it's the acidic medium, right? I have taken acidic medium, right? Now let's place a amino acid over here, carbon, COOH, over here H, here NH2, and here R. So as I said, it is a acidic medium. It means that H plus ions will be there. Some H plus ions will be here, right? Okay, what will happen, basically a H plus ion will go and form bond with this NH2, and they will form a structure like this: carbon, COOH, here H, R, and it will be NH3. And we know that NH3 is basically a base. It is base, right? So this is known as, this amino acid is known as basic amino acid. This is known as basic amino acid. So in acidic medium, the amino acid act as a basic amino acid, right? So consider that I'm having, oh my god, what happened? Okay, consider that I am having another container, right? And it is basically the basic medium. It is basic medium, right? So let's place a amino acid over here, COOH, here R, H over here, and here NH2. Okay, as I said, it's a basic environment. So here OH ions will be present. OH, 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 OH. Now what will happen, basically this OH will bond form with this H, and they will release H2O, like dehydration condensation will take place, right? And they will form a structure which will be known as, like, it will be CHCOO minus, here NH2, and here R. So this area is known as acid. It behaves like acid. So this amino acid is known as acidic amino acid. This is known as acidic amino acid. So basically in the basic medium, the amino acid behaves like acidic, and in acidic medium it behaves like basic, and it neutral, its behavior neutral, or you can say this is Zwitter ion, or it is bipolar, right? I hope this is clear to you. Now we are having, like, we have classified amino acids into 3 groups. Number 1 is non-essential amino acids. Second are essential amino acids, and number 3rd are semi essential amino acids, right? Okay, what does that mean? Non-essential amino acids which are produced in our body, are known as non-essential amino acids. They don't need to acquire this from our diet, from the outside source, so any source. They are synthesized in our own body. That's why they are known as non-essential amino acids, right? And they are basically 10 in number, right? Okay, then we are having essential amino acids. Okay, essential amino acids are, okay, I'm sorry, these are, yeah, okay, it's correct. Yeah, it's correct. Sorry, 10 are non-essential amino acids. Okay, essential amino acids which are not synthesized in our body, not produced in our body, and we need to acquire them from our diet because they are quite necessary, and our body want this, and we can't produce these amino acids in our body. That's why we need to have them from our diet. So these are known as essential amino acids. Basically they are 8 in number, right? Third, we are having semi-essential amino acids, means amino acids which are made in our body, which are synthesized in our body, but in quite lower portion, right? Synthesized in our body, but in low proportion, right? Which are actually, they are 2 in number, right? So I hope this concept is also clear to you. Now let's talk about proteins. Okay, first of all, let's talk about protein in the form of structure, right? First of all, we are having this primary structure. Basically, how are these arranged? These are like simple straight chain. They are straight chains, right? For example, we are having insulin. So in insulin, basically there are 51 amino acids, right? And there is one alpha chain and one beta chain. So in alpha chain we are having 21 amino acids, and in beta chain we are having 30 amino acids, right? So I hope this primary structure is clear to you. Second one is secondary structure. Okay, what will happen? Okay, basically we are in secondary structure. We are having two types of structure. Number one is this helix structure. This structure is known as alpha helix structure. Basically they are bonded by hydrogen bonds. Okay, second we are having is like the other secondary structure is something like this. Okay, these are known as beta pleated. This structure is known as beta pleated structure, right? And these are also attached with hydrogen bond. This alpha helix structure you can easily see in alpha keratin, right? And you can see this beta pleated structure in the fibrin of silk. fibrin of silk. Basically that's the structural protein. Okay, basically in generally we can say that the almost like generally secondary, like this structural proteins are secondary proteins, or even though our fibrous protein is also secondary protein. Okay, I hope this is clear to you. Okay, third we are having, which is tertiary structure. Third is tertiary structure. Okay, what is this? Okay, when these, like these alpha helix are joined to each other like this, in this condition, right, this is known as tertiary structure. Actually they are bonded by ionic bonds, they are bonded by hydrogen sulfide bonds. In tertiary structure we are having hydrogen, okay, over here we are having disulfide bond, disulfide bond, disulfide bonds by van der Waal forces 2. And this, like this secondary protein, it has hydrogen sulfide bond, it has hydrogen sulfide bond too, and also hydrogen bonds too. This is quite important for MCQs, the hydrogen bonds, right? Okay, that's tertiary protein. Next, okay, mostly the functional proteins are tertiary proteins. For example, are enzymes, these are mostly tertiary proteins. Myoglobin. Now what is myoglobin? Basically myoglobin is for example we are having hemoglobin, which like a pigment, or you can say, yeah, a pigment which is present in our muscles to in order to absorb oxygen. It's known as myoglobin. So these all are tertiary in structure. Now fourth we are having is quaternary structure. Quaternary structure, right, in which what will happen, these four tertiary proteins will be there, these four tertiary proteins will be there, right, alpha, beta, beta, these four will be there, and these are attached with each other by hydrophobic interactions, right, these are attached with hydrophobic interaction. I've seen this MCQs many times. They are asking that a quaternary structure are joined by, like they are joined by hydrophobic interactions. Okay, its most important example is hemoglobin, right, it has, like the alpha chain has 141 amino acids, and the beta chain is having 146 amino acids, and you can calculate 141 plus 141, it's quite 576, something like that. Okay, this is hemoglobin. Now if we talk about antibodies are known as immunoglobin, right, so antibodies are functional proteins. Basically these are functional protein, but they are made up of globular proteins. They are made up of globular proteins, right? So, and one more thing, in order to, for the MCQ's purpose, that in first, like in the process of protein synthesis, which amino acid is used first? So the answer will be there is methionine. methanoin will be the answer. That the first amino acid which is used in the protein synthesis is methanoin. So I hope this is clear to you. If you're having any kind of question, you can ask me.